Welcome everybody to the September edition of BioStock Live from Stockholm, Sweden. Today we have an exciting program for you. We have presentation from 4 Swedish biotech and medtech companies, and they're all focusing on a wide range of indications such as oncology, cardiovascular disease, Alzheimer's disease, and sepsis. We at BioStock also want to bring attention to the fact that today also happens to be World Sepsis Day. For this occasion we have invited a special guest, Ulrika Knutsson from the Swedish Sepsis Trust, who will shed some more light on sepsis. It's such a serious condition that kills millions of people around the world every year. And Ulrika will tell us something about what's being done to fight this global challenge. So first up, I'd like to introduce Ulrika Knutsson from Swedish Sepsis Trust. Welcome.
Thank you. Yes, thank you. I'm going to talk about Sepsisfonden. I'm a co-founder of that trust, but I think I'll start with showing you a little movie that we created when we launched Sepsisfonden in 2015. We realized at that point that one of our major issues or things that we needed to focus on was to actually explain what sepsis is. Maybe many of you know what it is, maybe someone has small knowledge of it and others more, but we realized that for the public to understand what sepsis is and to be able to actually raise the awareness around it, we needed to find a way to explain this rather complex condition. So we made a little movie. See here. Okay. How can I get this going here? Oh, there. Sorry about that one. Let's just watch this movie first and I'll come back to it.
Sepsis, formerly called blood poisoning, is usually triggered by a local infection, such as pneumonia in the lungs, an infected wound, or infection of the urinary tract. Should bacteria from the local infection spread or secrete toxic substances into the bloodstream, the immune system may sometimes go into overdrive, causing inflammation throughout the entire body. You get sepsis. This interferes with the blood flow, which leads to a drop in blood pressure, making it difficult for the blood to carry oxygen to the body's major organs. This can lead to failure of organs such as the lungs, the kidneys, or the heart. In the worst cases, the patient dies in a matter of hours. Detecting sepsis early is crucial, so be attentive to symptoms such as fever and chills, breathing difficulties, diarrhea and vomiting, confusion and slurred speech, low blood pressure and elevated heart rate, low urine production. Sepsis can affect anyone. Help us stop sepsis.
Yes, I'd just like to say that this movie is on YouTube if you want to use it in any matter and to explain what sepsis is. You feel free to do that. It's just write in what is sepsis and this will be on the top. Now, so this is what sepsis is, and how common then is it? Well, in Sweden, around 40,000 people develop sepsis every year, and out of these, about 20% dies. So it's a very high mortality condition. And it's also more common than the 4 most common cancer forms together combined, prostate, breast, skin and colon cancer. About 27,000 people suffer from those diseases every year. So it's a very common disease, and yet it's very unknown to many people. Some important aspects of sepsis. As I said, it's a high mortality. It's the most deadly acute condition you can have in hospitals today. You can compare it to myocardial infarction, which has about 7.5% acute deadliness. Early detection is really crucial, as also said in the movie. And this is because time is really of essence. If you detect sepsis early on and get the right treatment, it's more likely that you have a good outcome, as every hour is important when it comes to treating sepsis. And one aspect that we also have tried to focus the attention around is the link between sepsis and the rising problem of antibiotic resistance in the world. Because if we don't have antibiotic that can treat sepsis, it will be a really huge problem. And we can just look back about 100 years ago when the Spanish flu was a big problem in this world, and about 50, or at least 50, maybe up to 100 million people died of the Spanish flu. And the real cause of death when you died from the Spanish flu was sepsis, and that this was obviously before antibiotics was available. So this is a problem. And also in the war against sepsis, getting better methods for diagnosis and treatment, better treatment, is really crucial when it comes to fighting sepsis. I'll get back to that in a while. Now, so before we started Sepsisfonden, we did a survey trying to get knowledge around exactly how bad was the situation around awareness in Sweden when it comes to sepsis. And the blue line is in 2015, this was before we started Sepsisfonden. The awareness was as low as 21%, which is really very low, and especially compared to other conditions or other diseases. We gave questions around all kinds of diseases. You can see them on this side. Well, not the English-speaking people, but still. And as you can see, Swedes in general have good knowledge about our common health diseases, but sepsis sticks out. We did a new survey last year in 2017, and as you can see, it has increased to 30%, which is substantial, but still it's far too low. And that's why we're still working mostly with raising awareness around sepsis, because that's the most important task right now for us. And on the other hand, there's not any very low funding for sepsis research. This is actually numbers from the States, study from the States, but it's quite equivalent to the Swedish conditions. As you can see, sepsis is the most common condition, and this is the funds going into sepsis research. So that's also a problem. So sort of these two aspects of it made us start Sepsisfonden. So we want to increase the awareness and spread knowledge around sepsis, about sepsis among the public, but also politicians, decision makers, make them aware of it, and also within medical staff. And the other one is to raise and give out money to research that can improve methods for diagnosis and treatment of sepsis. That is why we started Sepsisfonden, and that's what we still work on. And our goals is to increase the awareness. Now, this is a number that we have aimed for 2019. We hope it will be more than that, and I'll get back to you why we think we might get further. We want to contribute to lowering the number of deaths caused by sepsis. We also want to contribute to getting better treatment for sepsis sufferers, and last but not least, to help survivors, sepsis survivors, to get longer and healthier lives, because that is also a problem. Some people get sepsis and get full recovery, but quite a lot of people have problems for a long time, and sometimes lifelong problems. Obviously, you can get amputated and stuff like that, but it also can be other things that you might not see on the outside, but like some quite common aspects of the rehab of sepsis is that you can get brain fatigue, depression, all sorts of cognitive problems. And these are things that is not really addressed, at least not in Sweden, as an issue for sepsis survivors. So that's also something we like to work with. So with that background, we started Sepsisfonden in 2015, September 3 years ago. You can, if you want, go to Sepsisfonden.se and read more about us. We are part of an international network. We work and collaborate quite closely to, obviously, World Sepsis Day, which is today, but it's also an organization. It's the name of an organization, World Sepsis Day. They work globally with these issues, and they have a really good website you can go to too. Global Sepsis Alliance is also, we are part of that, and I missed to put that in, but this May, also a European Sepsis Alliance was adopted. So we are part of that. And we also work quite closely to our equivalent in the UK, the UK Sepsis Trust, who have come quite far with raising awareness in sepsis, around sepsis in the UK, and we try to do things similar to them to make the same progress here in Sweden. Now, last year in May, WHO adopted a sepsis resolution. And this was actually quite good for us because we have felt a good push for us nationally too about sepsis. In short, this resolution elevated sepsis to a world health problem. And I didn't say that before, but in worldwide, about 30 million people suffer from sepsis every year, and around 7 to 9 million dies. So that is basically one person every 3 seconds dies of sepsis around the clock. So this is a worldwide problem. But when WHO addressed this and made it into resolution, it also increased the pressure on the member states to actually take action and an ownership of this problem and spreading awareness among public and also about health professionals. And interesting in this resolution was also that they identified the increased awareness as the key in the battle against sepsis. And the reason is that if people don't understand what sepsis is, and if they suffer from sepsis, they get it. They're sitting at home and having these problems, or a family member has it, and you don't understand what it is, then you maybe not seek healthcare or help in time. So since the time is crucial when it comes to treating sepsis, awareness is also crucial. So there's an expression that World Sepsis Day invented, which is really good. Sepsis is the #1 most preventable cause of death in hospitals today. And that means that a lot of deaths from sepsis wouldn't have to happen if we had the right knowledge in the public, but also in the healthcare system. We try to communicate. When you come to the hospital and you might think that you have sepsis, ask the question, could this be sepsis? Because quite often, it's even when you come to the hospital, they're not quite on their toes when it comes to sepsis. And it can take sometimes too long, 1 or 2 hours before they realize what it is. Sometimes can cause death because you don't get the right treatment in time. So increased awareness will lead to fewer people dying, and that is obviously the main focus for the awareness campaign that we are trying to now. We hope in 2019 to be able to do a national-wide awareness campaign. And we are right now talking to different kinds of people, organizations that we try to do this, collaborate with, to do this really broad, to the public, national awareness campaign. So yes, that was what I had to say today.
Thank you very much, Ulrika. Very interesting to learn about this big challenge.
It is
global
a big
challenge.
Yeah, it is.
So Sweden you have measured the awareness here,
Yep.
and we have a low yet increasing, thanks in part to your great work, level of awareness of sepsis here. What level of awareness is there globally?
Well, the problem is pretty much the same all over the world. Actually, the UK, much to the UK Sepsis Trust, has raised until 60% now. So they are probably the country in the world that has the highest. So that gives you an idea.
Yeah.
But quite a lot of countries don't have, especially in the third world. And in the third world, actually about 60 to 80% of the deaths are caused by sepsis.
Yeah.
Due to, hygienic problems and stuff like that.
Yeah.
Young children and pregnant women, this is the most common death in those countries. But they don't understand. They don't have the knowledge of it.
Okay.
But even in the Western world, it's a big problem that people don't understand what sepsis is, the knowledge is. And as I said, 100 years ago, people knew sepsis because it was everyday kind of, we formally call it blood poisoning, and that's, I might just address that. We have, in Sepsisfonden, made it clear that we want sepsis to be the expression, because blood poisoning is not an adequate, because sepsis is really like an umbrella, just like cancer is an umbrella for like 200 different diseases. But it's when an infection gets severe in the whole body. That's when sepsis occurs, and when the organs are suffering from the infection.
Yeah.
Yeah.
So you mentioned UK have a 60% rate awareness. That's really good then.
That's really good. Yeah, it is.
What are they doing? Is it a matter of funding, or is it something else?
I think they have worked a lot with national awareness, and they have actually had a lot of UK politicians are much up there and talking about it. They even talk about it in the parliament. I have some clips of that, and I won't show you now, but it's more a topic. And I think that's what we need to work with. Obviously work with the public, but also with our politicians to make them understand and make them raise the money that we need for, especially when it comes to the medical, the hospitals, to have the right skills and now we have some good examples in Sweden, like in where I come from in Skåne, we have Region Skåne, have sepsis alert alarms since almost 2 years.
Yeah.
And I know there's a few Stockholm. Both Karolinska and SÖS has started with that too.
Yeah, yeah.
And that's a good start, but we need to also on vårdcentralerna, whatever that's in, the local kind of health centers, they also need to have the same kind of knowledge.
Karolinska actually sent out a press release today stating that in 1 year, from September last year, their sepsis alarm has gone off, so to speak, 470 times or something like that. And they could see that the difference in people getting treatment with antibiotics differed by 3 hours for the ones that had these sepsis alarms and that didn't.
Yeah. So you see, that's I can just tell you also there's in October now, there's a clinical test going out in Lund concerning a blood test that could be like a quick test for an early detection of sepsis, because there's a protein that has been shown in research in a group of researchers in Lund.
Okay.
Could be an early detection for sepsis. So if that gets working, you can actually, whenever people comes into the ER, you can just check for that and get treatment going much faster.
Yeah.
So that's really interesting to follow and see what happens there, because that could be really
interesting. That's in Swedish.
Yeah, it is
in the forefront.
Yep.
So awareness is really important, and early detection or diagnostics is a key element as well.
Yeah.
And then, of course, treatment is crucial when you actually get this problem.
Yeah. Quick treatment. And
what's the lookout at this point for better treatment, so to speak?
Well, I think if we can raise the awareness and get more funding to the research, there's quite a lot of good research going on in Sweden and globally too, but there's just too little funding. So we have to realize, we are actually looking a lot on cancer research for the last 50 years. We can do a lot of things similar to that, as long as we get some more money. Well, not we, but the people researching around it. And I think there's a good lookout if we just, as a society, realize this is a problem that we need to address.
Yeah, the differences in funding that you showed earlier was actually quite shocking.
It is shocking. It is,
considering the prevalence of sepsis. But given then that the awareness is rising and hopefully funding will follow, at this point, why is there such a low interest or still in finding new cures? Or there aren't that many companies working on sepsis. In Sweden, for example, we will hear one of the few here today. But
I think the problem is just what we talked about. Cancer has been known for many years as a big problem for a huge group of people in the society. In the 90s, HIV was big. So we have to start talking about sepsis and the real problems with it. And I think when we start doing that and get the awareness not just to the public, but to everyone, then it will also be more hot to go into research and fund that and raise money for that. I think it goes together. If we have more awareness, then all the companies will also realize this is something. There's a lot of people that could be helped. So I think it's really a matter of awareness.
And there's this issue about different areas that are not equally hot, so to speak, for companies to go into.
Exactly.
Like now, immuno-oncology is really hot, for example. But what about on the academic level then? Because they're perhaps not as sensitive to trends, so to speak.
No, I think there's actually quite a lot of researchers want to do more, but the problem is it costs money to go into really deep research when it comes to sepsis. Well, into any research really. So I think there's a will academically wise, but there's just needs more money.
Yeah, and
especially to go into like the clinical state, as you say, finding now the test now in Lund. There's a Scottish company that has gone in with the money to develop that test.
Okay.
From the research done in Lund. So it's
they see the potential.
Yeah, exactly. They see the potential. And I mean, worldwide, if this really goes well, it could be a real hit worldwide. And for them, obviously, a good investment.
True blockbuster.
Yeah.
As far as Sepsisfonden goes, there is in Sweden. There's a huge actor within cancer, Cancerfonden, with the Cancer Fund, which every year contributes lots and lots of money to research. And is the goal of the Sepsis Fund to become something similar?
Well, yes. It might take us 50 years. I don't know to go that far. But yes, definitely, we want to. This is one of the things that we have been working for these last few years. Obviously, we are still in a financial sort of working our way up. We still haven't been able to actually give out money to research, but we now have some good. Well, I can't say really what, but we have some finances that we might have coming in. Okay. And also, if we can have a national world campaign, awareness campaign, we also think that we, from the bottom, can. Get more money, and in that way, quite soon give back to research. But obviously, it'll take time to go as far as the Cancerfonden, because they've been working for a long time, and they have. We are like me and Adam, and we're a small group of people working with this at this point, mostly.
Every journey starts with one.
Exactly,
step,
we have to start somewhere.
Yeah.
But that is our goal.
Yeah,
definitely.
Right. And if people who are watching this would like to contribute to Sepsisfonden, is there like a number for that?
Yes. Well, I should have had this here. If you go into Sepsisfonden.se, you find our Swish and our bank. Yeah. Yeah, and please. to contribute if you want.
Yeah. Are you doing anything special apart from being here, gracing us with your presence today at World Sepsis Day?
Yeah, we have quite a few people now out on different places in Sweden. Sepsis survivors who are handing out flyers and talking about sepsis. We have one on Svampen later on, and here in Stockholm on SÖS, and different cities in Sweden. On Sunday we have a sepsis relay, Sepsisstafetten, down in Skåne, raising money for and people running for this. And also today I'll be going over to Cereno to talk a little about Sepsisfonden and also giving away, we have a prize, Sepsispriset, that we give out once a year.
Yeah.
And this year we have given it to Otto Kars, which I don't know if you know him, but he's well known in the infection parts of Sweden, whatever you call it. Yeah,
infections.
He started. He was a very pioneer, really, when it comes to the awareness around antibiotic resistance.
Okay.
He started in '95 with Strama, which focused on this. And as I said before, this is something that we like to get people to understand the link between antibiotic resistance and sepsis. So that's what we're giving him the prize. So that's what we're doing around World Sepsis Day.
Okay, great. Thank you very much, Ulrika, and keep up the good work. It's really important, of course, and it was lovely to have you here.
Me.
Yeah, thank you.
Thank you.
I should say that BioStock actually is contributing in a small way as well. Global Sepsis Alliance that Ulrika talked about earlier. Today on the Sepsis Day, they have something called a Superhero Challenge, and people around the world do really tough things, challenges like swimming and running and stuff like that, and then they donate money for each mile or each distance they run and so on. And actually, the chairman of BioStock is participating in this challenge, doing a swim run with a friend of his. So they're gonna swim swim and run 16 kilometers in the Gothenburg archipelago in the end of September. So it's going to be really cold in the water as well, and this will raise some money. It's possible to see this challenge on Facebook if you just search for sepsis or a Superhero Challenge. I think you can find it, and please go in there and contribute, because all the money goes directly to the Sepsis Alliance for research and heightening the awareness. I think that's a great cause. Next in our program is actually one of the few in Sweden actors or companies that work with sepsis, trying to find a solution, trying to find, basically way to treat sepsis and acute sepsis, and that hopefully will be better than using just antibiotics. So I'd like to welcome Mikael Lindstam from Aptahem. Welcome, Mikael.
Thank you, Jonas. Good morning, everyone. Thank you for the nice introduction. Nice to be back at the BioStock scene. And I'm here to talk about awareness, which the previous speaker did today. And we all focus on a lot of awareness, but we also focus on treating the syndrome. So we think us have a very good candidate for a therapeutic solution. More about that later. This is just a forward-looking statement, of course. It's a mandatory for a public company, so I have to have it there. So we have been talking about the World Sepsis Day today, and I will not go into much. It will be already too much information, but I would say that we are sponsors of the Sepsisfonden in Sweden, and we're also supporters of the Global Sepsis Alliance, which is situated in Germany. And of course, we work our butts off to really address awareness, because the sooner you get in, the better chance you have to survive a sepsis occurrence. And if you don't, Aptahem will be there for them that needs it. Upcoming events in this awareness area, we have this today, of course. And as Jonas just recently mentioned, we have the Sepsis Awareness Superhero Challenge. And we will be part of that challenge, this 10-mile run swim in the Gothenburg archipelago, as Jonas just mentioned. And in November, we have an event that is from the medical faculty in Lund, Malmö, which is also the Sepsisfonden will be involved in, and we will be there and take part in that event. So we're looking forward to report from that. So Aptahem, yeah, we are public, I just said that. And we are a smaller biotech company. We are semi-virtual. We have some personnel, but we're not a big company. We hire a lot of expertise into the company. It's a very good way of running companies, especially in this country when funding and sponsoring is not that generous, let's say like that. Anyway, we specialize in aptamers and aptamer-based drugs, and we're looking at this at targeting inflammatory sicknesses in the acute domain. And that's where we want to be with the sepsis, because it's an acute syndrome that occurs. And we have a lead candidate, Aptahem, I just mentioned that, and we are pretty far with that one, and we have very good results in the preclinical models, I should say that. So we are very hopeful about having a cure against sepsis in later stages. In the early stages, of course, you use antibiotics and so forth. And we also consider ourselves having a world-class portfolio in knowledge, in patents, and in results that we have so far. And of course, we wouldn't be able to have this if we didn't have a fantastic team. We have collected a lot of fantastic experts in the company, from the cradle to goal, when it comes to development of a new drug. So I'm very happy to have that team around me. And this is the team, few faces on the team, management team. And as I said, we have a total coverage from start to goal when it comes to develop a drug. And we also have the scientific backgrounds, knowledges, and so forth, startup companies, running companies, deal making, license making, etc. But I want to mention a few people here. If you look at the upper left, it's Luisa Jedlina. She is the scientific officer of Aptahem, and she is actually the reason to Aptahem started once. She found the pathway, the commercial pathway for using the fantastic unique group of aptamers that we have, which are drug candidates. So she actually set off the company. It was her knowledge in immunology and in parasitology that made it happen. And later, in the upper right, Anders Bylund came in as chairman of the scientific advisory board, and he's also the CMO now. This year he became that. And he was the one that took it from top down. He looked at what would the market need and what should we do to get there. And then he split that down. And he came into the company in a situation where we saw new results from our lead candidate. We saw that it had a character that we couldn't really expect, and that was the anti-inflammatory character, what we could see there. And putting that together, and Anders put some thought to it together with Luisa, and they said, let's try it in the toughest inflammatory model there is, sepsis. So we did, and the results were better than we could imagine, actually. And that started a new strategy for the company some time ago. And Jonas already said it, we are probably the only company in Sweden working with the therapeutics to address sepsis syndrome. And I hope there will be others, so we could collaborate or, competition, healthy competition, of course. And it's again about awareness and also do something for this area. It's a tough area to be in. Many pharma companies did a lot of effort, but without having success getting a drug on the market. So it is a tough area, and that has to do with the multitude response what the sepsis infection gives you in the body. It's not a single occurrence, it's many occurrences. I will come back to that later. We are one of a handful in the world as we see us. I mean, there's a lot of academic efforts going around. We know this, and I will come back to that. We have collaboration with one of those efforts in the world. And of course, Big Pharma is focusing quite a lot now. I have meetings with Big Pharma that are very interested and have a new belief in that could be some drug candidates to actually address this tough syndrome. And that makes us pretty unique as we see it. I hope others also see that. And in combination with our first-in-class, let's say, matter, which is our aptamers, which is a new type of chemistry molecules, we have a really strong platform to stand on. And these aptamers I've been talking about, how did we discover them? This I could talk all day about. It's a very fascinating thing. You see the mosquito on the left side. And this injects malaria parasites into the bloodstream of the human being. And to survive in the human being, it creates a protein to bind red blood cells. for survival, and that's how it works. And this parasite is a master. There's no better biochemist in the whole world than a parasite. They can optimize the chemistry, adapt to the surrounding inside the human host. So it's have been optimized through generations to just sit there and do its job, and that's fantastic. I mean, this is so optimized, you can't find a better producer of a molecule. So we looked at this protein, we wanted to release it, we wanted to dissolve it, so we came up with a group of aptamers. And aptamers are built by blocks to form this structure that you see to the right. So you can make a perfect build around a certain molecule that you would like to inhibit. So we have made a blocker molecule or some kind of an antibody type of molecule. So what we are doing here, that we make an anti-molecule of this, so we inhibit further coagulation, as we see it, when the protein binds these cells. So that was the first, and we have evidence, and that's for the patent we have. And we used that to very good effort when we started the company as an anticoagulant drug, and it had excellent results in this area. But as you know, we made a strategic change some time ago and went into sepsis when we discovered that the anticoagulation was driven by the anti-inflammatory mechanism. So there we are today. So it's a combination, it does both, which is quite unique, I must say. But if you look at the structure, it's quite big, it has a lot of sites where things can happen. And as you see, there's anti-inflammatory, immunostimulating, anticoagulant. We see all this when we measure from animal studies in preclinic. It's quite fascinating to see, it's hard to believe. Some people are very tough to believe how it works. But we have a fantastic molecule, or a group of molecules that we work with. So, I was being an Ulrika before me probably talked about sepsis, and sepsis is a syndrome or an occurrence of something. It's actually initiated by an infection. It could be virus, bacteria, very common, as you treat it with antibiotics. It could be fungi, other things. It can be a cancer patient that just finished the treatment, and you're very vulnerable to the surrounding area. You have high counts of blood, white blood cells, which makes you very susceptible for other infections. So it's very common that cancer patients die from this. Heart patients have infections. You have lung embolies, whatever. So other sicknesses give in a second step sepsis. So that's important to remember. It's not the first thing, it's a syndrome that occurs. And the syndrome is when the body totally go haywire. I mean, it's a systematic inflammatory response in the body. The whole body is affected. And Ulrika mentioned this, that the most common way is that the body is reacting, it's counteracting, and that does it by lowering the blood pressure. Lowering the blood pressure means you get less oxygen to the vital organs, like kidneys, liver, heart, brain. And that leads to a dysfunction, a multiple dysfunction of organs, and that leads to death, sadly. Because if the organs can't work, you die. That's the sad story here. And yeah, I've already been talking about that we did a strategy change, and we fought hard and long, and when we saw the initial results of our lead candidate, we realized that we have a chance here to address the cascading process, what the systematic inflammatory response means. So, and also the fact that there's no real treatments out there. So, I mean, it's a big need in this area, and I hope more will come in. So I welcome competition, of course, because this is about the awareness and then how you treat this. And I'm talking about imbalances and systematic conditions here. I just want to show here two different scenarios. The left scenario shows what happens in the body. I've talked about the multitude of mechanism here. And we have the anti-inflammatory response, we have the pro-inflammatory response, we have the immunosuppression. And that throws everything off. One thing's happened here. Body rushes over to try to take care of this incident, but before it does, the whole scenario changes. So actually the positive way of the body thinking, taking care of something, actually becomes a negative thing for the body. And this is why you have this total chaotic, uncontrollable situation in the body, where you have a lower blood pressure, you can have massive coagulation, leads to massive strokes, or you can have massive bleedings. The body totally loses the control. Body is very good on regulating everything, but when it can't keep up with this very fierce and fast infection, it goes totally imbalanced. And that's what we want to show here. If you then look at the right side, that is what we see or a theory about what Aptahem does. And of course, our preclinical results have shown and supported what we have here, that we counterbalance this. So we go and inhibit these processes happening because it has a multi-purpose. So if you just had a drug that didn't do or only do one of these inflammatory parts, like anti-inflammatory response, you would still have the other parts to address. So we address the whole problem as we see. And the results speak for themselves in preclinical that we actually do this. But more about that on the next slide. And we have very nice results. And we start with improved survival. That's the key, you know. That's the ultimate biomarker. Will the patient survive? Yes. So we have significant survival. We have also dose response when we do this. So we can dose up and see better efficiency when we dose. And that's very important for the medical situation, that you can play and analyze the patient and do optimal treatment for that one. And we're talking about repeated dosing. We have made dosing at different time points during this occurrence of sepsis infection, or stimulated as such. And we see that we have significant survival during that time, even later in the stage, which is a very, very good indication indeed. And that means also we can dose more than once and twice. We can keep and follow the patient to make an optimal, at least the scenarios like that, an optimal treatment for the patient and save the patient. And we already talked about the restored blood circulation. This is key. And when we do our preclinical studies in animal models, we see that the blood pressure is withheld. It doesn't change. So with Aptahem, we see the blood pressure is kept. And of course, with the blood pressure kept, we have oxygen in the system, meaning that the organs fare very well. And we see that on the typical biomarkers that they use in a trauma unit or a hospital or whatever you do, typical test you do if you have a suspicion that you have some infection, we look at the same markers. And when we look at these markers in our studies, we see that they are back to normal status or better than the group that didn't have Aptahem. So we have a lot of evidence in these preclinical models speaking very good for the case of counterbalance and get the body into right balance and get. Safety profile. We have seen through the years that Aptahem fares very well in different systems, and we have made acute tox studies to show that we have a very high ceiling. And with that, we always kept with at least 10 times margin when we dose animals, just to keep it in a sane area. And just currently, we are doing the preclinical toxicology program, and we will report about that later. But that's the real toxicology program. But still, we have a feeling where we have been from the beginning with acute tox. And a little about our program. You have probably read, some of you, that we have just announced some delays, and I will try to describe where we are right now. If you look at the two standing pillars, that's where we are at the moment. And then we start with the non-GLP toxicology study. It's finished in practical ways, but now we have the analysis, we have the documentation, the final reports, etc., etc. But it fared positively. This much we can say today. But we will announce, this will be an interim report. This is the first part of the preclinical toxicology program. The next part is what you see, that's called GLP toxicology studies. And that's the one that is the regulatory part of it that leads to a document that we will file to the FDA, EMA, or the Läkemedelsverket in Sweden to be approved to go into clinic 1 with Aptahem. And on the side, we do other things. We do mechanistic studies and so forth, but we also have a big NHP study, non-human primate, and that's as close as we can come to humans. So this is why it's interesting for us to study Aptahem in this case. And we recently announced that we will prolong this due to that we saw very good tox results. So we want to expand and do higher concentrations, and this is to just see how far we can go and get the window for where we can dose. And also having more statistics, because anything that can be patented, we will patent, because that means money in the bank in the future. That's how it works. And that also increases the attractiveness for the company. I would also like to say another point up there. We need to make some additional non-GMP manufacturing, because we use so much in the non-GLP tox. We couldn't really expect that much consumption, but it happened. And as a final thing, we are doing a GMP manufacturing. The GMP is a good manufacturing practice. That means it's a regulatory thing. And we have to have GMP approved substance to put it into man. Otherwise, it will not be approved. So there where we are at the moment. We do other things as well. And due to that, we filed a new patent application last year. We can now be a little bit more open about what we are doing. So we have started a collaboration with a hospital in Seattle. Very interesting. And we are now discussing with a big European institute to do the same there. I have to come back to that later. Academic publications is on the way because now we don't have to be tight-lipped any longer. We are very happy that we can start public announcement of what we have so far. And there's a lot of other things around it, like mechanistic studies that we're doing. Just to show the market case, because if we don't have market case, no one will be interested to take us to market, so that's how it is. But you see the incidences here, top 5 European countries and United States. There are many incidences, and of course the mortality is very high, as Ulrika has spoken before about. We did a very modest estimate. We look only at the anti-inflammatory treatment, not the antibiotics. Just so you see the 32% in Europe and 11% in US. But anyway, we look how much each patient would cost and the scenario. And we're looking at the multi-billion US dollars per year, which is what's needed to attract the big drakes to come in and take a look at us. Okay, just to sum up a little, we think we have and we feel we have a very good expertise. We have a fantastic portfolio. We have results, we have patents, we have built the team from start to goal kind of setup, and we are very happy about that, of course. Pipelines, Apta1 you know now. We also started Apta2 as a second pipeline, and this is just initial studies, but it has Apta1-like properties so far what we tested. But we have to do a lot of more investigation before we can say more about this, and we will announce that. But it will be in the inflammatory domain, for sure, we can see that. And we have a third one, and all these are protected by patents. So all these are our main candidates, and these are, of course, potential pipelines, which Apta1 is the first one. We also look at a partnering license deal, etc. It's important for us to find someone to carry us through it because it's multi-million-dollar to bring us to the market. We will not be able to do this ourselves, of course. So I'm working very much with that. And of course, we can look to build value in the company and build the volume of the company by in-licensing or acquisition of new drugs, for example, just to build more momentum into the company and to make it more secure and not being seen as a one-trick pony. And we're already not a one-trick pony, as we have 3 potential pipelines already. And just to summarize, I will not say so much. I mean, I've said it all already, but we have a strong foundation, and it's resting on a novel drug candidates in form of the aptamers. And I think I will hold there. Thank you very much.
Thank you, Mikael.
You're welcome.
Very interesting to see this development going on in this area that's in such a big need for a new therapy, basically. If you should sum it up, what makes your case interesting as far as that it could be a solution for an acute therapy?
Well, there is no drug on the market today that do what we can do with this. At least the indications are like this. I'm pretty sure there's a lot of things going on in academic sections, so we know this. This is why we will have a collaborations with some of them. But there's a renewed interest because as we have a novel type of chemistry, I mean, the hopes are up again that there is something that can actually address this in a way that Apta1 does because it has a multi-effect. So I think that's crucial that's what it is, yeah.
Mm. And being then one of the few actors in this field, I know you were at the Nordic Life Science Days earlier this week and probably had some meetings there. What kind of level of interest do you see when you speak to?
Well, there are those who are very interested in this area, of course. It's for the bigger players like pharma, it's a strategic decisions. Either you do or you do not. And you can have the most interesting profile. It doesn't matter. If they're not strategically interested, you will not look at it. But there are quite some that are looking into this field, as I just mentioned during my talk. And there is an interest, and it's an interest for the aptamers' behavior, the profile, not only for the case of sepsis, because we see that it treats a lot of inflammatory responses in the body, the most crucial ones in other areas too. But we've chosen to go into sepsis because it's such an awful syndrome. So we choose to do that because it's so powerful, our aptamers, and that's why we choose that. But we have talks and discussions about other applications with this aptamer because it's so multifaceted in its behavior.
And if you're familiar with biotech and drug development, you know that things take time. But you were still you were forced to announce that you had a bit of a delay recently, or are looking at a delay. Can you tell us something about what happened?
Yeah, I mean, despite all good measures and efforts that we do, and we now announce timelines, these things happen, sadly, for most. Yeah, we had a case here of that we used up all our drug for the toxicology studies, and we need to make more. And then we had some challenge with the GMP. It's a totally different thing to make GMP. It's surrounded by a lot of regulatory things, and you have to be very precise when you do this and keep a lot of things. And we haven't reached the purity levels that we need, as we need to put it into man, and also to get the approval from the authorities. So I mean, but our own goals are very high. So that's what we're working now. So yeah.
And this problem is being handled?
This is being handled right now, of course, yes. So we are moving forward all the time.
And speaking of moving forward, and we only have a short moment left, but what do you see as the most important upcoming milestones for you then, for Aptahem?
Well, we have to finalize the talks, the total talks. I mean, now we do the first half, and then we do the second half, the GLP study, as you saw in the presentation. And when that's done, we summarize everything, and we put a file together, go to the authorities, and ask for them to look at it. We want to do this. We want to shoot it up in humans and test it for safety in humans. We want to see, do they get black spots, or do they get delirious or something, or they just feel fine. That's good. Then you're fine to the next step. But that's what it's all about. I think that's a milestone. And of course, these smaller milestones now to crack the purity thing. We have an NHP study that I think will be very interesting when we conclude the results from that round. We may have new patents around the corner, publications, other collaboration, very important collaboration, I would say, to aid our own development. It will be very important, and we're so happy that we can be more public about it now when we have been tightly for all these years. And I feel sorry for the market, you know, waiting for more information all the time. We want to tell you everything, but you know, the market and everything about patents and so forth puts them on us to be quite tight-lipped. It's very frustrating, trust me. I would like to tell you as much as possible. That's how it is.
We're looking forward to an outspoken Aptahem coming up then.
Are we not already?
Yeah, even more so.
Okay.
Thank you very much, Mikael.
Thank you very much.
Thank you.
Thank you.
So, for our next speaker, we will leave sepsis. We're still going to be in the area of blood, I could say. We're going to listen to Sten Sørensen, who is CEO of Cereno Scientific. Well, they come up with some interesting news today. I'm sure we're going to hear more about it right now. Welcome, Sten.
Thank you. Good to see you all. I'm very pleased to be here. You know, I'm the CEO of Cereno Scientific. We are a company that is involved in developing a first-in-class prevention drug for the major killer in the planet, thrombosis, blodproppar in Swedish. And I have the great news to tell you. This morning we released a press release that we got a patent approved on the largest pharmaceutical market in the world, the US market. And thrombose prevention therapy is around $10 billion in sales every year. So that's a key milestone for us to get that patent approved. But we'll leave that, and I'll get back to the story of Cereno and what we are about. So thrombosis is, you probably have family or friends that have experienced a heart infarction or stroke, or even a blood clot in the veins and so on. So it's very common, and it's the #1 killer on the planet. The sales around the world is around $20 billion yearly. Not only those who suffer a thrombosis in the heart or the brain or elsewhere are suffering from this disease. There are 10 times more people that survive than actually are killed by thrombosis. Every second there's somebody getting thrombosis. Now, current drugs are used to weaken the coagulation system in the body. We have the coagulation system to stop bleeding, so that's a good system. And the blood thinning drugs weaken that system to prevent thrombosis. And that's what we have today on the market. And the problem with that therapy is that because it weakens the coagulation system, it puts everybody treated on an easy to bleed state. So there is a risk of bleeding, severe bleedings in the brain or in the stomach or elsewhere. And not only severe bleedings, but also any kind of bleed. So around 3% of those who get this therapy get severe bleeds, and up to 25% of those treated get any kind of bleed. So bleeding is an issue, and cardiologists know about this and are worried about it, but they need to treat thrombosis. But risk patients don't get therapy. Those are at increased risk of bleed. And because of this, the therapies are developed in moderate dosages, so they don't have the optimal effect that you would like them to have. And that's why one treated patients in 5 years get a new infarct or new thrombosis event. So that's 20% in 5 years that get a new event. So there is a problem in the market of bleeding, the risk of bleeding, and would like to have more preventive effect, more efficacy on the market. So, and that's is exactly what Cereno is addressing. Now, so what's used on the market? Let's look at this axis. You see it's like a line there. It's a balance line. And the body is, often works like that. So one system, the coagulation system, is working to heal a wound. And the fibrinolytic system is actually taking care of blood clots so they are not growing too much and becomes an occluding blood clot in the vessel. And they are normally in balance. But if they're out of balance, we are at higher risk of developing a blood clot. And what you can see on this line, to the left, there is a number of therapies used for preventing thrombosis. And they're all working to weaken the coagulation process, thus the bleeding risk. Now, Cereno is working with the body's own defense mechanism against blood clot. We are working to restore our own defense mechanism. We have such a system. So we have, if you think about this room, and there are sprinklers for fires, and they start to sprinkle water when there is a fire in the room. That's how the body is defending itself against blood clots. The body has sprinklers, so to speak, in the vessels everywhere. And if there's a small wound in the vessel, it will sprinkle out a fibrinolytic dissolving substance called TPA, and thus stop that blood clot from becoming too big and occluding the vessels. Cereno is working to strengthen that defense mechanism in humans. And let's look at how that can be illustrated. So this is a vessel that's cut in half. So we look into the vessel, and when we get older, we get atherosclerosis and we get plaques in our vessels, and they're a bit stiff. And when they crack, there is a bleeding. And so it starts to bleed, and then there are signals that go up to the wall of the vessel, the endothelial cell wall, and there sits the sprinklers. And there is a substance there that will be released by this bleed. And that's the fibrinolytic substance, the defense mechanism. And it will make sure that the clot is not too big. There is another protein in the blood floating around called pi. And that is supposed to take care of TPA when it's done its work, or if there's too much coming out of the sprinkler. So this is the normal defense mechanism. Now, the problem in risk individuals for thrombosis is that that system is weakened. It doesn't work as it's supposed to be. And that is the case if you are hypertensive, high blood pressure, if you have diabetes, if you're overweight, obese, and so on. So those individuals have a higher risk of developing blood clots. And one reason is that their system of defense is weakened. And if you look at the left side here, you can, that's illustrated by a heart. And the coronary vessels are supposed to give the heart blood and oxygen and so on. In the normal circumstance, if you have a plaque that bursts, you see there's a small blood clot forming, but then TPA is sprinkled out and take care of dissolving the clot so it doesn't become too big. And pi, the blocker of TPA, is in low concentration, illustrated by small letters. If you look to the right, there is very little TPA coming out of the sprinkler because there isn't TPA in the wall. There is too little stored by this person. And in addition, the blocking substance of TPA is too high concentrations. And you might know this, that in the morning hours, it's most common to get myocardial infarction, also stroke. And that's also when pi, that substance, the protein, the blocker, is high in concentrations in the blood. Now, what is Cereno doing about this? Well, we are working, let's jump back. Don't know. Here I should be able, sorry. So we are working, and we have found that our substance, we call it CS1, our drug candidate, can reverse the situation from the right here to a healthy and much stronger defense mechanism in the body. We will, if you take our substance in advance, that will improve the synthesis of TPA, so it's stored up in the cell wall, so it's ready there if there's a bleed. And it will also, if you have this therapy, reduce pi, so it's not in high concentrations. Yeah? The beauty of this system, and it's why it's first in class, nobody else has done this. And by pre-treating, we are not weakening the coagulation system. We are not causing the patients and putting them at risk of bleeding. We are just pre-treating to restore the proper defense mechanism of the body. So it's not our agent that is dissolving the blood clot. It's the body's TPA that's doing it. And we have made sure that it's there in the sprinkler. So we're not putting patients at risk of bleeding, and we're having a positive effect. And the body activates its own sprinkler when needed, by itself, not done by us, so to speak, or our substance. So we are moving ahead, documenting our substance, and our aim is to document the preventive effect against blood clots. And as a first-in-class, we will do this. And it will be a valuable alternative to what's out there on the market. And everybody is looking for an effective drug that doesn't cause bleed. Now, let's look at some documentation. So this is from an aorta in a mice. And they've been treated with the controlled substance and with our active substance, VPA. And the green there is TPA, the water in the sprinkler, so to speak, the protein in the sprinkler. And you can see if they're treated with our substance, they have much more TPA. So they are ready to take care of blood clot. This is in live animals. This is in mice. And they have been treated for 3 days or 96 hours. And the graph to the left shows those who have been treated and those who were controlled. The treated is the red line. And these mice were hurt, so there was a bleed in the vessel in the stomach, and there's a blood clot forming. And as you can see, the red line is much lower than the black line. And that's documenting that it's preventing fibrogen buildup, or the blood clot buildup. And already after 2 minutes, you can see they separate. So there is a preventing effect of blood clots appearing in the mice, in live mice. And to the right, you see a photograph of the thrombosis formation. So we have documented that this works in live animals. We've also been in man and documented the release of TPA and the reduction of pi by 50%, the protein floating around in the body, which is a main risk factor for myocardial infarction. Well, do we have any other evidence that this might work in man? Yes, in 2011, a Danish research group published a study on the whole Danish population. And they looked at what epileptic drugs were used over a 10-year period, and they looked at cardiovascular incidence. Were there difference between different treatment groups? Yes, in one group there was a difference. And so this is a major study, 150,000 patients. And in one group, the group treated with a substance called valproic acid. That's our substance that we are using in our development. There was a 40% reduction of myocardial infarction in this epileptic population. So it looks like this is preventing heart infarctions in man. There has been other studies. An Irish study came 2014 in 20,000 patients, same data. And actually this year there were 3 studies published with similar data, preventing effect in stroke this time. So we have developed this substance, valproic acid, into a specific innovative controlled release formulation that is targeted to take care of the pi, the floating protein in the body, that is a risk factor, and also to make sure that the TPA is synthesized and stored in the vessel wall. And we have brought this substance into man in a phase 1 trial, and we published the data in June, and it showed good safety and tolerability as expected, because it has been in man for 45 years, but not used in this patient population. We also looked for if this formulation also reduced pi. We wanted to show, because it's a specific formulation to take care of pi in the morning hours. And it did. So we have a significant reduction of pi. Very positive data for us. So we are developing into phase 2. We are in phase 2 now. And when you want to develop pharmaceuticals, you need to finance it. And phase 2 trials are expensive. So we financed the company with up to 106 million Swedish in March this year. We published the agreement, and it's a specific agreement where we can pull down funds as we need them. We did this financing agreement because we needed to secure the funds over a long period of time. It has hurt our stock, but the risk is reduced for being able to get the work done. And when we have a phase 2 study done, I'm sure that people will be happy with the valuation of the company. So, new first-in-class drug, innovative formulation, short-term to market, has a double mechanism of action: increase TPA synthesis and reduce pi. That's what we are about. So we have not said that much about our development program, more than the latest press release this week, that we have signed an agreement with the CRO. It's OCT based in St. Petersburg, Eastern Europe, and offices also in the US. We have done a procurement for over 6 months with a number of CROs, and we've done feasibility where it's best to do this trial and so on. And we ended up signing the agreement with OCT. And we will do the study in 20 centers approximately in Russia and Bulgaria. And we aim to submit the documents for approval for the study by the end of this year. What else? This is our development line. I should also mention that when gearing up for phase 2 trials, you need to increase your capacity in different ways. So we just announced this morning that we have assigned a new chief medical officer in Björn Dahlöf, associate professor from Gothenburg, Sahlgrenska University. very well published and experienced professor from Gothenburg. He's also a major shareholder in the company and sits on the board. He now goes in operatively to work with this trial, amongst other things. He's been involved in many trials globally. We also then today released that we have gotten our patent approved, or notice of approval for the US market. So this has been a very good week for Cereno. And I hope you understand the impact that this drug will have if we succeed. I've been fortunate to be in development processes a couple of times that have changed medication globally and improved survival. And I think this drug has a possibility to do something similar. All right, thank you.
Thank you, Sten. Very interesting to hear. A huge indication you're working with, a huge market, of course. Would you say, what are the biggest challenges you're up for?
Yes, I mean, if you have great ideas, there's lots of challenges. You need to be able to tell the story, and you need to create followers that believe in your story for different reasons, for competence, capacity, for financing, and so on. And that's a long road. As I mentioned, I've been doing this a couple of times before in major pharma. And even inside a company with lots of billions, it's a road that you need to travel. So I think in our situation, there's always a scientific proof and documentation that you need to do. And we have so much competence. People involved in the company were hired and developed thrombosis medication, prevention medication for AstraZeneca that are global sellers today. So we know what we're doing, we think. Yeah. So we actually think that the financing is the major challenge for this program. And as I mentioned, that's why we took this financing, despite that it also have some negative impact short term for us. It's not the only financing we will do based on our strategy, but we needed to do it. So
it's a means to an end.
Yes.
You mean? Yeah. Speaking of talent and competence, at the Nordic Life Science Days conference, industry conference that was the other day, that was a recurring subject, I think, to the difficulty it's the biggest challenge in Nordic life science companies, basically, to attract talent and to find the right competence. However, as you mentioned, you announced today that you have a new CMO, and you didn't have to go that far to find him, did you?
No, I mean, and that's actually fortunate for us. I've been working most of my life in cardiovascular medicine, and here or abroad, but globally. And fortunately, AstraZeneca, which I used to work for, my first job in '84 was for Astra, but I came back in '95 and worked globally. But AstraZeneca is one of the leaders in the world in research and has been since the '60s. So we have lots of talent to pull from. So that's fortunate. And we actually moved our company into BioVenture Hub, which AstraZeneca has opened up for smaller companies that they think have a great future and promise, and not necessarily conflicting with what they're doing. Yeah, so we have talent that we can bring in. You always want to bring in, when you develop drugs or you're on this path, you'd like to bring in the best people in the world. You need them, and you can do that in phases. But because Astra has been so good at developing drugs and has been exactly in this field, we can bring the best in from a local perspective, so to speak.
Well, that's convenient. And you mentioned yourself the financing perspective, another challenge, of course. you have a solution in place now. You're looking at next step of development, and you're going into a clinical phase 2 trial, and looking to do that later this year. And that adds on, you're going to another level then in clinical phases, and there's costs related to that. Are you financially set to go into that phase, or?
Yeah, we are. So, and again, that's why we did the agreement we did. So, the whole procurement process has involved actually defining almost down to the bare bones the details of the study we will do. So that's all done, except for the parameters in the work that needs authorities' approvals, and there's some work left there. But the whole agreement with the CRO is based on a very detailed plan, both in objectives, activities, and contents and measurements and so on, and patient recruitment times and all of that. So it's time and events based. And I should maybe mention that we are doing the study will be done in orthopedic surgery. It's a great model for the pharmaceutical industry and has been in the past to prove proof of concept in man to prevent thrombosis. And so you replace your knee. And there are 15,000 knee replacements every year in Sweden, for example. So that's a model that we will be using in measuring this.
And in terms of the next clinical phase, then what patient numbers are you looking at to include there?
We haven't released that.
Okay.
But
you can tell me.
Yeah, I am not allowed due to the stock market regulations. But it will be, there are 20 centers. So it will be multiples of the hundreds. So it's going to be it's a trial that the big pharma would do. Yeah. Okay.
And then going forward, it's going to require, providing everything goes as planned, you're going to need to do even bigger studies, next coming phases. And the strategy there is to find a partner who can help you do that, or?
Well, somebody said, I was at a CEO forum for listed companies within this field, and somebody said that who'd been very successful in making a deal recently, a major multi-billion deal in Swedish kroner this summer, and said that their philosophy was that we'll always tell the world that you'll do it yourself as long as you can, and then whoever is interested to come and be partner with you, they're welcome to discuss. And I think that's our philosophy. We'll move with this concept until the world understands how good it is. But normally after phase 2, if you have very good data, that is a valuation inflection point for the valuation of the company, normally, a project or a company. If you pass phase 2, you have proof of concept in man, normally. And that's when big pharma or partners want to go in, because they want to form the phase 3 program themselves. But we might pursue a phase 3 ourselves too. So we haven't, we're open.
Okay. We're looking forward to learning more about that in time. Thank you very much for coming in today.
Thank you. Yeah. Thank you.
So from one huge indication to another huge indication, which affects many people around the world, and I bet some of you have been personally affected as well, close by, and so I know I have. And it's a pleasure to introduce Per Wester, CEO of Alzinova, who are working to find treatment for Alzheimer's disease. Welcome back.
Thank you very much.
Yeah.
So, thank you very much for inviting me coming here. I will today tell you about an oligomer-specific active immunotherapy, which is a quite new invention in Alzheimer's treatment. So, it's important to understand that the evaluation and the development of new drugs in Alzheimer's disease, everybody says it's a very tricky one. But in the last year, there has been two major breakthroughs. And they are both within the amyloid beta field, which is very promising also for us because it's exactly the same field as we are working in. So you can see that aducanumab, for instance, which is a monoclonal antibody that is owned by Biogen and they have a cooperation with Eisai, has got very far in the phase 1 clinical trial that they did and moved directly to phase 3 with that compound. And on top of that, we had some very, very interesting results this summer. So in July, there was some presentations about a phase 2 study with a monoclonal antibody named BAN2401. This is an interesting compound because it was invented here in Stockholm by a company named BioStock. And they have also shown some very, very interesting results. And when I say interesting, I mean really clinically benefit for the patients. They have improved cognition measurements. Maybe not so that they get smarter, but the deterioration is going down in a slower pace. So this is something which everybody thinks is very, very interesting. These two drugs are the only ones so far that has been showing some kind of clinical relevant effect. And it's also interesting to see that many people on the stock market believes that there is a 65-70% chance that these drugs will actually reach the market after the clinical work has been done. So that's very, very promising. So we are moving away from these small percentages up to double digits and more than 50% chance of getting registration. That's fantastic. And Goldman Sachs says that Biogen is worth maybe 12 billion peak sales estimate with aducanumab. So it's a huge opportunity. There is no question about that. Okay, it's very important to understand when we look at this amyloid beta field that there are different forms of amyloid beta. They are not all toxic. So here, for instance, we see this monomeric form, which can be found all over the body. If you find drugs that target these, they will be very occupied finding these species throughout the body. And when it comes to available antibodies available to do whatever treatment it needs to do in the brain, there is not much left. So that is one very, very important factor. And we can also see that the monomeric forms are transferred to oligomeric forms and also fibrillic forms. The fibrillic forms are shown as plaques in the brain. And they are more or less some kind of reservoir of amyloid beta. You should leave them alone. That's the current message. This one is the real bad guy, as seen here. So only oligomeric forms of amyloid beta are toxic. What they do is that they sit on the neurons and block the transmission of synapses so that the neurons are dying. We see cell death, brain cell death. That's more or less what is happening. And just a short comment on this one here. It is very, very soluble, and it moves from the soluble form to stable form like this one, and also go back to this one. Here and here, they are not toxic, but while they are here, they are toxic. So this is a tricky one. So what Alzinova has done is very interesting, because we have developed by some bioengineering. You see this C30, C21 binding together two cysteine bridges, which is making it impossible for these monomeric forms to become fibrils. They are locked as oligomers. As you can see here, no fibrils are formed at all, so we can skip that problem. And stable oligomers for drug development can be used. And that's exactly what we have done. So we are developing a unique disease-modifying active vaccine for AD. So these are the oligomers, A-beta-42, that's our synthetic oligomers, which we actually put in a syringe and inject into, this case, animals. We have done it in mice, in rabbits, and in non-human primates. We have seen strong response on immune response on all these species, and we have seen very, very clearly almost clean of side effects. But that's not very unusual when it comes to animals, because they cannot complain about being nauseous or having problems with the knee or things like that. So, but this is still very, very promising. So the next thing now is to move over into man, and that's also the plan that we are preparing for now. So we have now non-toxic oligomers specific antibodies created in monkeys, rabbits, and in mice, soon to be tested in man. That's what we are doing right now. So we are more or less finished with the preclinical phase and moving over to the clinical phase very soon. So let's see where we are in this field of amyloid beta development programs. It has been a huge work from many companies, putting in a lot of money. As you can see here, are all the big pharma companies who has been investing billions and billions of dollars. And the first generation was mainly focusing on binding to this A-beta-40, A-beta-42. The problem is that they can be found all over the body. So if you have an antibody which this antibody recognizes, could be then an A-beta-42, for instance, monomeric form, they bind there and they are happy. They have done their job, mission accomplished for them. However, the problem is that, so these are occupied there. The problem is when the disease is actually happening in here, in the brain, so it's a brain disease. When these monomeric forms pass the blood-brain barrier, and also the production of monomeric forms, which are produced locally in the brain, they start to aggregate. And they form these fibrillic forms and the oligomeric forms, which we talked about 5 minutes ago. And because the fibrils are much more in quantity and the oligomers are much less in quantity, the antibody that binds unspecifically to aggregated forms, they will easily find much more fibrils, which are not the dangerous kind. So what happens is that they get some signs of clinical benefit, which we have seen. And they get some side effects in form of this ARIAS, the brain swelling, which is well-known side effect when you release amyloid beta from the plaques, where they are stored stored in the brain as reservoirs. So what Alzinova has done is developing this specific antibodies which only binds to the toxic oligomeric forms. You see, it doesn't bind to this blue one, the fibrils, only the really toxic ones. And for that reason, all of them will only bind if they find this target. And they just circulate and circulate until it finds the target. And this will soon be evaluated in man. Again, coming back to, there has been two very, very interesting results with BAN2401 and aducanumab, which are two antibodies which has been showing some clinical effects already. They are very interesting because they work on the same kind of target. But as you see, ours is a little bit more specific to the oligomeric form, not just the aggregated form. And we have now a monoclonal antibody, which we call ALZ201, and we have an active vaccine, which is called ALZ101. And the ALZ101 is our main product that we work with to develop for the market. This is quite interesting. You saw some interesting results this summer. BAN2401 from BioArctic presented data on a very, very large patient population, 850-something patients. And it has been investigated. This was back in 2010, investigated the binding profile of this monoclonal antibody, MAB158, which is the precursor of BAN2401. So this one is the same antibody, more or less. This is a mice antibody, and this is the human antibody, but it's the same kind of antibody. And we did a test and looked for how it binds the antibody MAB158 to our oligomers, our synthetic oligomers, and versus how it binds to the wild type oligomers, the A-beta-42 wild type oligomers. And as you can see, it's very clear that it's twice as high affinity to our oligomers than it is to the wild types. So this is interesting because BAN2401 have a clear effect. We know that now from the phase 2. And it's binding very, very strongly to our oligomers. So this gives us a hinge that it may well be that with this very powerful oligomer, it will be possible to have some strong results in the future for treatment in real patients. But however, we haven't seen that yet because we haven't tried yet, so we need to do that first. Okay, treatment of Alzheimer's patients is a challenge, and the reason is clearly described in this figure. If you think about a timeline, and you think about cognition function, and you have your career as an Alzheimer's patient, the first 15 to 20 years, you have your disease, but you don't see any clinical effect. You don't start to forget things, or you're living quite all right. But after this period of time, there comes a point when you move into this, which is called MCI, mild cognitive impairment. And then that is when you start to get your symptoms. And you let just a few years more go, and you will fall down to full dementia. And as you even further go down, you cannot live anymore because the brain is falling apart and your brain is dying, so you get clinically dead. And the possible options for different treatments can be seen here. So if you put in, for instance, a monoclonal antibody here in a patient that has gone very far, what you can do is to increase the length of the rest of their lives. So they get some more months or maybe some more year to live at the level that they are right then, and then still deteriorating from that. If you catch them earlier, you get a few more years, and you still also break the deterioration, so patients will not be able to fall down so deep and so fast. But the real opportunity lies in starting to treat patients early, before they have any clinical effects, but they do have the disease. And then you need to come in sometimes here during the 15-20 years. And this would not be possible with a monoclonal antibody, an antibody which is produced in the pharmaceutical factory. And the reason is that this drug is extremely expensive to produce. We have seen that also when they go off patent and there are biosimilars. The prices are not going down like with small molecules, like 90-95%. It goes down 10-15%, something like that. And the reason is that they are so expensive to produce. And on top of that, it's also interesting to understand that the monoclonal antibodies has to be given every 2 weeks. BAN2401, for instance, is every 2 weeks in a hospital ward. So you can imagine 50-year-old people standing in a long queue. Every second week they go and get their shot, and they do that for the rest of their lives. Normally, a monoclonal antibody treatment in other diseases will cost around $20,000-$30,000 per year per patient. So it's very, very unlikely that these patients will be given this kind of treatment early on. They will probably have some proof that the patient has become a patient, that there is a problem with the symptoms. However, if we have an active vaccine, which is much, much cheaper to produce, but also gives the opportunity to wake up the body's own immune response, so you don't have to come back every second week, but instead we believe every 6 months or 12 months for a booster dose. That gives us a possibility to use this period before we start to see deterioration. And this, you can see this line here is much longer, so it's much more effective to treat patients early before things go too far. So this is a difference. And I would say that this is also something that makes us different from example, aducanumab and BAN2401. So we don't really compete against them. It's not our competitors. It's quite the opposite. We really hope that they will make it. We really hope that they will come to the market. It will be the best thing for us that they can make it all the way and be quite successful, because that will build up the opportunity for us as well, but in a different segment. We are moving into the first clinical trial, which is settled to start around year end this year. And it will be done in Finland, in Turku. We call it Åbo here in Sweden. We have a synopsis in place. There will be 24 patients, mild AD, so they are actually patients, not healthy volunteers. Primary endpoint, of course, will be safety, tolerability, immunogenicity. So if there are any problems coming up, or if the immune system is kicked off, that's what we want to see. And exploratory biomarkers, CSF, tau, phospho-tau, those kind of biomarkers are interesting and important. And we also know that if that is something that can be seen, it's going to be interesting for the whole community. So I was telling you about an oligomer-specific active immunotherapy, which is a new innovation in Alzheimer's treatment. I hope that you have seen the innovation in our opportunity, that the oligomer is really the target, and it has to be specific in order to be effective in the future. And with that, I say thank you.
Thank you very much, Per.
Thanks.
Very interesting. This is a tricky area, tricky indication, as you mentioned, and billions and billions of dollars have been poured into this field.
Yeah.
And well, unfortunately, lots of backlashes in the last 5 years or something.
Yeah.
But is like a new optimism rising now with the recent advantage that has been occurring?
Yes, we see that. I think the people we talk to clearly see that. And that is, of course, driven by the success with the BioArctic and their cooperation with Biogen and ASI. We are very happy for their sake, but also for us and for future patients. It's the best thing that could have happened. And now we have 2 compounds having shown clinical effect, and clinical effect is the only thing that matters. That's the only thing that will be giving access to the market for these products.
And you mentioned that you're going towards clinic yourself, going towards proof of concept in man, and so mentioning this that you have now two compounds that has shown proof of concept, and does that give you like a really good confidence going forward?
I think most of all it lowers the risk in our what we offer. If there was nothing out there giving some kind of meaning to believe that the amyloid beta would be the one that could give an opportunity for the future, that would be a great risk. But given now that we have 2 recent clinical trials having shown that there are actually effects good enough, if they can copy the results in phase 3, they will for sure be registered. So they just need to show what they have already shown.
Yeah. And this you mentioned that you have your concept is more specific, actually. How important is that in terms of attracting interest from big pharma players, for example, who you will be dependent on at some point?
Yeah. I would say it's data driven. So if this is turning out to show that the theory shows to be correct in reality, meaning that if you have a specific enough antibody being produced, it will just skip all the things that is not toxic and bind to the toxic stuff. And there will be enough antibodies available to bind to this toxic stuff. If you have something which is not that specific, it will bind to all the irrelevant ones. So if they find some toxic, there will be no antibodies left over. So that's more or less is the theory behind it.
And it also has impact then on the possible side effects.
That's true. Many people say that the fibrillary forms, which means the plaques, are reservoirs of amyloid beta. If you hit them and bind to them, there will be release of monomers. And they are typically situated in the inside of the blood vessels. So what happens is that they give the blood vessels swell So it's called ARIAS, which is a typical side effect, and in more common language they say brain swelling.
Okay, yeah.
So, not good.
Nothing to wish for.
Yeah.
I have not much time left to ask you. I should say that you will be here afterwards, so
Yeah.
that people can catch you and
Yes.
ask you stuff.
Yeah.
However, I would like to say that then you said something about you're happy for advantages by, well, competitors, but you don't see them as competitors.
No.
Why is that? Do you foresee a future where you could combine the therapies?
Yeah, I think this is basically it. So I think that if the monoclonals will be here somewhere and ours will be here somewhere, I see a possibility that if you start treatment with our active vaccine and patients start to deteriorate, then it can be used in combination with other drugs. It might also be that we know, for instance, that when there is a flu coming in, it's much more difficult for elderly people to handle the flu than younger people, and that depends upon the immune response that every individual have. And older people tend to have less strong immune response, immune defense. So it may be that some of these treated by our treatment is not that effective because of this reason, which means that it could be possible for them to use other stuff.
But in general, early diagnostic would be beneficial, of course. Yeah. And just today, actually, there was news from BioStock that they entered a collaboration on diagnostics specifically with a Gothenburg-based company.
Yeah.
And
you see, it's happening.
Yeah, it's happening, and I would say that would actually benefit Alzinova as well. Then, if there was such an improvement in the diagnostics field.
Absolutely, I fully agree. It's good news.
And finally, then. Going towards the market, are there any specific possibilities for a drug candidate like yours in terms of time to market, seeing that there's such a huge need?
Yes, and I am actually a member of a group with the Swedish authorities, and it's more or less reflecting the regulations that EMA has. And they have something called prime status, which means that you can get fast track through EMA regulations. And FDA has something else called fast track. You can get some kind of adapted stepwise approval. So you can start to use a drug in certain patients and register everything you see. So this would, for instance, be possible here very early, because otherwise we had to do clinical trials lasting for 15 to 20 years, which is obviously not possible. But if we do it in this way, that would be clearly an opportunity. So.
Well, definitely looking forward to following your progress. Yeah. And thank you very much for coming here today.
Thank you very much for inviting me.
Yeah.
First time for me here.
Yeah, that's great.
Yeah, very cozy and nice. Thank you.
Yeah. So our program is running towards its end, but we do have one more company who will present themselves today. And we're swiftly moving from one big indication to another, and now we're going into oncology. Spectracure is a Swedish medtech company based in Lund, and they will tell us today about how they plan to, or how they actually are treating prostate cancer. So, welcome, Johannes.
Thank you. Thank you very much for that introduction. Good morning, everyone. So I am the CTO of Spectracure. So I work with the technical and the clinical development in the company. So let me just also mention our CEO, Masoud Kayami, who has been the CEO for the company now for, I think, about the last 7 years or so.
So some words about the introduction of Spectracure. We're working with something called photodynamic therapy, PDT. It's a combination treatment where you use a light-sensitive drug and combine that with laser light. And you only get a therapeutic effect when you illuminate the tissue where you have the drug. But we're a medical device company, so we don't actually develop the drug. We use a drug that's already available, but we develop the laser system and the dose planning tools that are needed to perform this treatment. And we focus on the treatment of prostate cancer. So our company has now been around for about 15 years. It's based on research at Lund University that's been carried out for a long time, more than 25 years now. There has been more than 3,000 treatments done using PDT on superficial tumors. But the problem with PDT is that when you try to apply this on internal tumors, such as the prostate, it's difficult to get the light into the tumor because the light has a very poor or short penetration depth in tissues. So that's one of the main challenges of Spectracure and which we have solved. So this is the board of directors of the company. So our chairman is Ingmar Kihlström. He has a background in the pharma industry. He's been working for some of the large pharma companies in Sweden. Masoud, our CEO, is on the board. And we have 3 of the founders also on the board: Sune Svanberg, Katarina Svanberg, and Stefan Andersson Engels. And these are presented on the next slide. So here they come. So Katarina Svanberg is now, she's retired from clinical work, but she's professor of oncology at Lund University. Sune Svanberg, her husband, is professor of physics at Lund University. And Stefan Andersson Engels, he was professor of physics at Lund University, but he's now moved recently to the Tyndall Institute in Ireland. We're working with a lot of external collaborations, consultants, but the main partners we have are the hospitals that we work with in our clinical trials. So these are our partner hospitals. We have the Skåne University Hospital in Lund, of course. We work with Princess Margaret Cancer Center in Toronto, University of Pennsylvania Hospital in Philadelphia, and University College London Hospital in the UK. And we're right now running a phase 1 clinical trial for locally recurring prostate cancer. And that trial is run at these 3 sites: Princess Margaret in Toronto, Penn in the US, and UCL Hospital in UK. The trial is, as many of you probably know, is up and running in Toronto. We've done a number of patients there. And we're just about to start treating patients also in London. And we are planning to start up patients also at Penn in a very, very short time. So before I go on describing where we are here, I will give a little background about PDT and how our treatment works. So PDT is a triple component treatment, actually. You need a drug, which we call a sensitizer. You need light, and you need oxygen. The oxygen is always present in the body, so that's not something you have to supply, but you have to supply the drug and the light. And when you have all these 3 combined, you will get tumor death. So next up is a little movie of how the treatment works. So this shows the anatomy of the prostate. The yellow blob in this picture is the prostate. We use a template that guides needles into the prostate. And this is done by ultrasound imaging. So the surgeon can, during the procedure, see exactly where he's putting the needles. These needles are then guide these optical fibers into the tumor, where we can guide the laser light. So this this is supposed to illustrate that the cancer tumor is killed. But the point is that we illuminate the laser light through the optical fibers. But we can also do measurements, perform measurements through the same fibers. And that is needed to perform the dose calculations that are needed to get the correct treatment. So this is a slide that shows what the treatment looks like. This is from Toronto, one of the treatments we performed recently there. The first slide shows treatment planning. The surgeon has to know where to treat, where the prostate is, where to put the needles. And we have developed a certain set of algorithms and the software for doing that. We call it iDose. Next up is the needle placement. This is done with ultrasound guidance. And you see the surgeon putting in the needles through this mechanical template. And in the third picture, you see the actual treatment going on. Those are the optical fibers that are led in through the needles into the prostate. So what the status in the trial. So it's a phase 1 trial, clinical trial with locally recurring prostate cancer. And we've treated 6 patients so far. And the purpose of this trial is to establish the correct drug and light dose. So we're escalating the dose to see until the point where we get a clear effect and we don't see any significant side effects of the treatment. So establishing the correct drug and light dose and evaluate the safety, those are the purposes of the trial. We have previously shown some preliminary outcomes. So we have, seen some very positive results on the patients that have received the high dose of the drug. So to the left, you see an MRI of the prostate before the treatment. So within the red circle is the prostate before the treatment, and to the right you see the prostate after treatment. So essentially we have no blood perfusion through the prostate after treatment, which is an indication that essentially the tissue is dead inside the prostate. So why are we looking at recurrent prostate cancer? Well, we see that as a strategic route to the market. So we're not focusing, at least not now, on primary treatment of prostate cancer. There is a clear clinical need because these patients who have recurrent prostate cancer, they don't have any good treatment options today. You cannot typically treat them with radiation again. You cannot do surgery on them because the tissue has been so compromised from the first radiation treatment that it's very difficult to perform surgery on these patients. So typically they only get hormone treatment, which is not a curative treatment. It's something that can prolong the life of the patient, but it also has some significant side effects. And this is also a large patient group, so it's it's an influential patient group. So these are the reasons why we're focusing on this indication. So that leads us a little bit to the background, the prostate cancer market. So there are about 600,000 men each year that are diagnosed with prostate cancer. And of that group, approximately 1/3 will get external beam radiotherapy. And from that group, approximately 1/3 again will have a recurrent cancer. And that's the group that we're looking at now, where there are no routine curative treatments on the market. And the hormone therapy that's typically done has some significant side effects. Then if you look at the other treatment options, the big one is surgery, of course. About 50% of all cases will have surgery. And if you don't get that, you might just wait. You have active surveillance is one option. Or you can have one of the other treatment options which are not really that common. So the technology that we have, we call our system the Spectracure P18 system with iDose. So it allows to use up to 18 optical fibers that are put into the prostate. And there's one laser connected to each of these optical fibers. The system has an optical switch that allows us to switch between treatment mode and monitoring or diagnostic mode. And that's something that's unique for Spectracure. We have several patents that cover this technology. We also have this iDose software for treatment planning and for dose monitoring. And there are several parts of that that are also covered by several patents. So that's also very important for us. Some of the advantages of using this method is that it's a minimally invasive procedure. There's no open surgery involved. The recovery is fairly quick. It's a repeatable treatment. Unlike traditional radiotherapy, you cannot repeat that, but our treatment is repeatable. There is a low risk of severe side effects connected to the drug. The drug that we use is already approved worldwide. It has a very well-established safety profile. And there is no ionizing radiation involved in the treatment that could also potentially be dangerous. The system technology is relatively low cost. I mean, the manufacturing cost is relatively low. If you compare it, for example, the machinery that's needed for radiation therapy, which is relatively expensive. Some of the market background. So the total global market for prostate cancer treatment and prevention is about $50 billion today, and it's expected to rise to $63 billion in 4 years. There are more than 2,000 clinics in the US that offer a similar type of therapy called brachytherapy. Brachytherapy is also a therapy where you insert needles into the prostate, but in that case you insert radioactive seeds into the prostate rather than the optical fibers that we use. But sort of the logistics and the practical way of doing the procedure is similar in brachytherapy. So that will make it, we think, easier for the hospitals and the clinics to adopt our technology, because they don't have to make a big leap into some new technology. There are a few treatment options for recurrent prostate cancer, and there is an active. The health authorities are actively seeking new treatments. There is also a trend in prostate cancer care of quality of life and not only survival, because the traditional treatments, radiotherapy and surgery, they are associated with some side effects that are not very attractive. So our business model, we don't intend to sell the actual device. We intend to sell treatments, and the treatment package would then consist of consumables needed for the treatment, and that would be optical fibers and needles. It will also come with a one-time use of the laser device, and the laser device would then be provided either on a loan or as a lease or something like that. The drug that we use is called verteporfin. The trade name is Visidyne. The patent for the drug has expired, so in principle you could develop a generic version of it. The hospitals could either acquire that by prescription, or we could license it in and use it. Provide it together with our treatment package. So the initial market that we're looking at is these 60,000 patients with recurrent cancer in the US and the EU. We're looking at the cost of this treatment of about 35,000 euros, and that would make this total worth of this market about 2 million euros. But this is what we aim to launch the product for, the recurrent cancers. But we're looking at the primary cancer market. And so if we look at this picture again, we would like to get this chunk on the right, where you have these patients that are now doing other types of treatments. and get a piece of that market as well. And that's a very big market. I mean, the recurrent cases are only about maybe 10% of the total. But if we could get to the other 90%, that would be a huge market for us. We have a number of patents that protect our technology. We have 5 patents that cover the hardware, the laser instrumentation, and so on. We have 2 patents for consumables. That's the sterile optical fibers and the needles. And we have 5 patents that cover the iDose algorithms and the software. And we have patents now that, I think the last one expires in 2032, and we have a number of ideas now in the lab that we are planning to file new patent applications for. So that was my last slide. So with that, I'd like to thank you for listening.
Thank you very much, Johannes. This is a very interesting concept and a very interesting market, of course, as well. Your clinical trial that's going on is that you have treated 6 patients.
Yes.
Is that right? How many will you treat? What's the scope of the trial?
So this is a dose escalation trial. So there's no fixed number of patients. It depends on how high we have to go in dose. Okay. But for the last patients that we've treated now, we think we are getting close to the final dose. Okay. The forecast now is that there will not be so many patients more to treat.
You see the light in the tunnel. So
yeah.
Sorry, no pun intended. And the patient group, you showed us how big the original group is, and then it funnels down to the recurrent group, and some do get hormone treatment there. So how big exactly is the subpopulation that you will be targeting? In the first.
So it's hard to get exact numbers here because the statistics are very variable in different countries and so on. Oh, yeah. But we're looking at maybe 50 to 60% of those 60,000 patients per year that we think could be eligible for our treatments. Okay, and competition-wise, you mentioned that there are no curative options. There's hormones and of course other options as surgery. So, but in this field that you're active in, what are you up against? Well, in PDT, there is no competition that does exactly what we're doing. I mean, this type of dose planning and laser systems that we have, that makes us quite alone in the market. There are companies working, pharma companies working with developing these kind of drugs that are used for the treatment. There was one phase 3 trial published, I think, about 2 years ago on prostate cancer in the UK that showed promising results. That was not for recurrent prostate. It was primary low risk, but still it showed that the method is viable for doing on prostate cancer. So we don't necessarily see them as a competitor. I mean, I think it's good to have a few players in this market that can show that it's a useful technology. That should also raise awareness and interest. And speaking of interest, and if you can repeat these promising results that you've seen so far, and well, you think that would ring a bell at the offices of some bigger players, perhaps? Yeah, I mean, we have 2 options for, continuing the development. I mean, one would be to carry through a phase 2 and a phase 3 trial ourselves and then go to the market. But I think a more likely scenario is that we will partner up with a larger player, either in the pharma or in the medtech industry, that will support the late development of this program. Yeah. And if that scenario plays out, are you looking for a specialty company, or are you just looking for the biggest player possible, so to speak? I mean, I think we're looking at the company with it should be a fairly big company, but with a good match for our technology as well. Yeah. Okay. And well, you actually very recently announced that you are planning on a financing round. Yes. Can you tell me something? Yes. So we issued a press release, I think, yesterday with this, because we're at the stage where we have to renew our funding. So there is now a suggestion for how to do this, but it is now pending that we have need approval from the owners, current owners, of course. But the plan is to take in about 52 million Swedish kronor during fall, and then there will be warrants for another 35 million next year. So,
okay. Yeah. And what will these funds then go to exactly?
Yeah, so we have funds now to more or less finish the phase 1 trial, but that will cover, the new funds will cover the phase 2 trial and then the start of the phase 3 trial.
Okay, so we'll take you quite some way along the road. And as of now, what would you consider to be the most important triggers or milestones coming up for Spectracure?
Yeah, so the most important one, of course, is to finish the phase 1. And by saying finish, I mean that we will perform the last patient treatment in phase 1, because then there will be a 12-month follow-up after each patient. But we don't have to wait 12 months before we can start the phase 2 trial.
No.
We only have to wait 1 month to see the acute possible side effects. After that, we can initiate the phase 2 trial.
Okay.
So that would be by the end of this year, maybe start of next year, something like that.
And will these trials be made, is it locally, or do you work with, is it international studies?
Yeah, so as it looks now, I think because we have now developed this very good collaboration with these 3 hospitals, I mean the Toronto, Philadelphia and London. So the plan is to continue the phase 2 trial at least with those hospitals, maybe add another one or so. It would be nice to work with someone more local here in Sweden. Depends on whether they are available. I mean, they might have competing trials and so on with this patient population. But
yeah. And speaking of that and competing trials, and sometimes it can be hard to actually, well, if there's not patient material enough, so to speak. How do you see it? Or do you see a lot of interest from participants? We
do, but it's always a problem that, I mean, there are lots of men with prostate cancer, but you have to fit into all these criteria that are defined by the protocol of the phase 1 trial or the phase 2 trial, and so a lot of them have to be excluded because they don't fit into the criteria. So I mean, enrolling patients is a bit of a challenge, and also because the actual procedure that we perform, we need to be in an operating room, and we need a lot of staff. I mean, we need nurses, we need doctors, and so on. And just planning that and make a reservation for a time where every one of these specialists can be at the same time, it's a bit challenging. And that's why we have been seeing some delays now in the trial. But it looks very promising now, and we have a number of patients now that are lining up in the trials that hopefully will be treated in a very short time. So.
Okay. And final question, then, given that you will get approval for this financing round, what would you say are the key components in Spectracure that would make it interesting to invest?
I would say that, I mean, we have shown promising results so far from the phase 2 trial. And that's one thing. And I mean, there is a very big market that we can look at in the future. So there is, I think there is a very big business potential in this project.
Yeah. Okay. Thank you very much, Johannes, for coming here today. And it was a pleasure having you and listening. I'm sure I will get questions both from the audience, but also my colleague Linus will do an interview with you later. Thank you very much.
Thank you.
And that concludes our program for today here at BioStock Live from Stockholm. Be sure to join us again next time. Thank you very much for watching.